This is the second part of your meiosis notes, and before we take notes, this is a little bit of a different video. What I want you to do is get a blank sheet of paper. It can be computer paper, it can be notebook paper, but it does need to be a longer piece of paper. You might even, if you're a big drawer, or if you want to have some extra space and not feel cramped, maybe even tape two pieces together. And what I want you to do is I want you to pause this video and I want you to draw what I have here. The blue dashed lines kind of divide your space into four parts. Try to keep them on top of each other. I had a little bit of an issue with my bottom left one kind of encroaching a little bit, but don't obsess over it. Don't, redo it if you think you've messed up. Just do your best to make sure you have space to write, because we will be putting chromosomes in these circles, because those circles are cells. So go ahead, pause the video, sketch this out, draw the circles, draw the cells, label them, and then join me again when you're ready. All right, so I'm going to assume that you have this all sketched out. We're going to start up here in the top left with interphase. And I know you can't see where my cursor is unless I write something, so I'll do my best to be as specific as possible. But what we're going to do is start with a cell, and we're going to assume that it's a body cell, so a skin cell or something like that. And we're going to go ahead and we're going to draw in 2 chromosomes, and I know that humans have 46 chromosomes rather than 2, but just for simplicity and just because the circles are not big enough, let's go ahead and draw only 2 in. So I guess we can consider this to be a hypothetical cell. And this cell, red is for mom, doesn't really matter, blue can be for dad, is going to be diploid, which means 2n equals 2. It's a very simple cell, so obviously not a human skin cell like I said before, but two chromosomes, 1 from mom and 1 from dad, with a diploid count of 2. So 2n equals 2. And during G1, this cell will grow. During S, the DNA gets copied. So go ahead. And, I also forgot to mention that you should have 2 different color pens so that you can differentiate the chromosomes. Remember, the chromosomes are going to make copies of themselves. And I'm not going to draw the nucleus. I'm not going to draw any organelles. I'm just drawing the chromosomes so we can keep track of the chromosomes, right? Even though the number of DNA has doubled at this point, because there is still only 1 centromere in the middle of the chromosome, even though these are double-stranded, they are still only considered to be 1 chromosome. So there's still only 2 chromosomes. We're still diploid here. Nothing about G2 changes the chromosomes. So you can go ahead and just redraw those chromosomes or carry them over. And that's it for interphase. So let's assume that this is a body cell. It's a somatic cell that's going to do mitosis. We're going to proceed to the right. So the first step in mitosis is prophase. So we've labeled that as P. There are, the nucleus is breaking, the nuclear membrane is breaking down, the chromosomes are condensing. Even though I've drawn the chromosomes out here in interphase, you guys should know that you can't actually see the chromosomes during interphase, right? They're not condensed during interphase, but just for simplicity and to keep track of them, we have drawn them that way here. So I guess formally, technically, now they would start to condense in prophase. So I'm going to go ahead and just carry them over again. Remember, nothing really different is, besides that, is different. If you have an animal cell, the centrioles are also starting to migrate, etc. But again, for this video, we're just focusing on the chromosomes. And then during metaphase, M, this is where things start to get a little bit more specific. The chromosomes line up in the middle of the cell. So I want you to, in the middle of the cell, go ahead and draw your 2 chromosomes. And let's also draw the spindle fibers as dashed black lines. And this is important, right? And I don't know where you might want to write this. You might want to write this kind of up below or up above or where you have space. But go ahead and make a note here that it's the chromosomes that line up in the middle of the cell. The chromosomes line up, right? The next part is anaphase. So during anaphase, I want you to make a similar note. I want you to write that the sister chromatids separate. The sister chromatids are what separate. All right, the sister chromatids separate. In telophase, the nuclear envelopes do start to reform, and the membrane does start to pinch in. But in terms of the chromosomes, they're set. They're on their side of the cell. And then that's the end of mitosis. And then you have cytokinesis, which is the dividing of the cytoplasm. And you have your 2 daughter cells. All right, so if you do a chromosome check, let's check the chromosome count at the end result here. So we have 2 daughter cells, and each daughter cell, that should be in black, but that's fine. We can do it in red here. 2n equals 2 for both of them, right? They have 2 chromosomes each, just like each other, and just like the original cell over here in G1. All right, so that ends cell division. If this were a body cell or a cell maybe doing asexual reproduction and doing mitosis. Let's say, for the thought experiment here, that this cell will produce gametes rather than identical copies of itself. So in this case, we're going to drop down to the bottom left. So here, again, we've gone through G1, S, and G2, so we're going to drop down and we're going to do P1. Remember that meiosis is the process where gametes are made, and there are 2 rounds of it. So we're going to go through meiosis 1 first, starting with prophase 1. And remember that prophase 1 is where you have crossing over. So I've deliberately drawn it so that part of the red and blue chromosomes cross over right there. Right, so make a note that during P1, prophase 1, that is when crossing over occurs. That's when the homologous chromosomes swap bits with each other, right? Then you go on to metaphase 1. The key here in metaphase 1, here's another note, is that the homologous chromosomes line up in the middle of the cell. The homologous chromosomes, the tetrads, line up in the middle of the cell. This is different than in mitosis, where the individual chromosomes are the ones that line up. Here in meiosis, the tetrads, the homologous chromosomes, line up, and that difference is absolutely critical. So we're going to draw this. See if I can do this in a way that's clear. And remember that the chromosomes have crossed over at this point, so I'm going to put a little bit of red on the end of Dad's blue chromosome here just to show that. and a little bit of blue on the end of mom's chromosome here, again, just to show that. Now look at the difference. This is a visual thing. Notice that here's my spindle fibers. Notice that in meiosis, or metaphase 1 of meiosis, again, the homologous chromosomes are lined up in the middle rather than the chromosomes themselves. So visually, just take a note of the difference between those two, right? Between metaphase of mitosis and metaphase 1 of meiosis. In anaphase 1, here's another difference. The homologous chromosomes are what separate. And actually, you know what? I have space down here to note this. So I'm going to go ahead and do that. Remember, this is a different thing than what happens in mitosis. So again, the homologous chromosome separates. So I'm going to draw mom's chromosome getting pulled that way. Dad's chromosome getting pulled this way. And remember, crossing over, has happened. So I'm going to include that little blue piece on mom's and that little red piece on dad's. Again, visually take a note of the difference between anaphase 1 in meiosis and anaphase in mitosis, right? Here, the sister chromatids. In anaphase and mitosis, the sister chromatids are separating. The sister chromatids separate, whereas in anaphase 1 of meiosis, the homologous chromosomes separate. Nothing different happens in telophase regarding the chromosomes, so go ahead and add them in there too. And really nothing different about the chromosomes happens in cytokinesis 1 either, so go ahead and add them there as well. And then go ahead and add them in prophase 2 also, because for the sake of the chromosomes, again, nothing really is different at this point. And remember, don't forget the little crossing over pieces. So I'm going to go back and put my, my red and blue sections in. All right, now by the time you get to metaphase 2, this process looks pretty much identical to mitosis. The chromosomes line up in the middle of the cell, and then the sister chromatids separate. So let's go ahead and draw that. Don't forget your crossing over bits. So again, just like mitosis, the sister chromatids are going to separate here in anaphase 2. Sister chromatids. They're not full sisters, remember, even though one is a copy of the other. By this point, there has been some crossing over. So they're not completely sister chromatids, but they are still referred to as sister chromatids. And they get pulled apart. So here we go. Let's have that one and that one. That one and that one. Don't forget the pieces, oops, from crossing over. Color that blue. Color that red. And, oh, you know what? I forgot the spindle fibers, so let's go ahead and add spindle. All right. And then in telophase, nothing has changed about the chromosomes. Remember, you have the nuclear membrane reforming, but the chromosomes are still going to look the same. They will start to uncondense as well. But for this, we can just keep the drawing the same. And then you have a second round of cytokinesis after mitosis 2 has finished. And you end up with 4 haploid daughter cells. So let's do a chromosome check. So here's our daughter cells down here at the bottom, or sorry, on the right side. And there's 4 of them. There's 1, 2, 3, 4. So there are 4 daughter cells. And if you look at each of them, this top cell, I'm going to call it number 1, has mostly mom's chromosome with a little bit of dad's from crossing over. Chromosome 2 is all of mom's. Chromosome 3 is all of dad's. And chromosome 4 is mostly dad's with a little bit of mom's from crossing over. So all of the chromosomes, or all of the daughter cells, have different chromosomes in them. None of the genetic material in the daughter cells is the same. It's all different. Not only are they all different from each other, but they are obviously different, if you go all the way back up here to G1, to the parent cell. Or sorry, to G1, right? They're all different from the parent cell. So not only are they haploid, but they are all different from each other and different from the parent. And there's 4 of them, right? If this were happening in a male, each of these 4 cells would be sperm. And you can draw a little flagella on these if you want, or you can just leave it alone. They're a little whip-like tail for the flagella. If these were egg cells, remember 3 of them would become polar bodies, and 1 of them would actually hog the cytoplasm to become the egg. Right. One more question though, at what point in meiosis did the cells go from diploid to haploid? When did we go from having 2 chromosomes in this example to having only 1? You can pause if you need to. All right, I'm assuming you've paused and you went ahead and looked. It happens after meiosis 1. So if you do a chromosome count, let's go to meiosis 1 and P1, right? There are 2 chromosomes here, 1, 2. There's 2 chromosomes here in, I'll write this here, there's 2n equals 2. There's still 2n equals 2 in metaphase 1. There's still 2n equals 2 in anaphase 1, but they're getting pulled apart from each other. And by the time right here, right after telophase 1, between telophase 1 and cytokinesis 1, look at the number of centromeres now. These are supposed to be connected here. You only have 1 centromere. You have 1 double-stranded chromosome. So you've gone from diploid to haploid, n equals 1, after the first round of meiosis. After the first round of meiosis. And then you stay haploid for the remainder of meiosis 2. And again, just to recap, the key points here is that, let's go up to the mitosis section. In metaphase of mitosis, the chromosomes are what line up. In anaphase, the sister chromatids separate. In meiosis 1, you have crossing over in prophase 1. The homologous chromosomes line up in metaphase 1, and the homologous chromosomes separate in anaphase 1. The sister chromatids don't separate until anaphase 2. So I hope this helped, and I'll see you next time.